Hello, everyone. I'm Lisa, your guide for this scientific presentation titled Cloning the Unclonable. We're thrilled you're here with your coffee, tea, or maybe even your pipettes in hand for what's going to be an insightful session. Today, we're joined by the brilliant Dr. Jessica Hartman. Her background reads like a scientific highlight reel. Degree from the University of Virginia, PhD from Duke, postdocs at both Baylor College of Medicine and Duke. She's led research and development across multiple biotech companies and now plays a key role in designing next-generation cell biology workflows at Cell Microsystems. In short, you're in very good hands. Before we launch into the good stuff, a couple of quick reminders. We are recording today's session, so don't worry if you miss a slide or want to revisit something later. We'll send you the link in a couple of days. We'll follow up the presentation with a live Q&A session. So send your questions anytime using the Q&A feature. With that, I'm excited to hand the virtual stage over to the star of the show, Dr. Jessica Hartman. Take it away.
Thank you, Lisa, for that super kind introduction, and I certainly hope I can live up to all that high praise. I'd also like to thank you all for joining me today to learn about some of the work that we've been doing to clone the unclonable and overcome bottlenecks in cell line development. So in today's webinar, what I'm hoping you'll take away from our conversation is a few key points that I've highlighted here. We're going to spend a brief time discussing cell line development and the need for mono versus polyclonality in your cell line of choice. We'll talk about some of the challenges in cell line model development, and then ultimately how we have been developing protocols to overcome these bottlenecks in our lab and in yours. So if you're joining us today, you're probably familiar with how canonical cell line development works, but to get us all on the same page, I thought it would be useful to kind of discuss some of the methods and methodologies people are using to do this. For the most part, when you begin to develop a cell line, you have your line of choice, and you have to introduce your gene edit, your genetic material, whatever you're trying to introduce into the cell, using one of the methods highlighted here. It could be something like lipid-based transfection, viral transduction, electroporation. But regardless, however you're trying to get material into the cell, the next step is then to create a heterogeneous population of single cell suspension in order to create a population that has your gene edit of interest. The way that you turn that polyclonal population into a cell suspension and into a single cell population varies depending on what you have in your lab, but it could be as simple and as manual as 96 well plate manual limiting dilution, or as complex as a 3D printer or a flow-based sorting system. And ultimately, the choice of how you enrich your population can really dictate your ending cell population and whether or not you need to do further refinement to create a poly versus monoclonal population. So you can see here in these two examples, in some cases you might just be enriching for every cell that has the edit, or in other cases you might be trying to create a pure population that is generated from a single precursor. And so when you're thinking about generating a cell line, the first question most people ask themselves is, do I need to clone or do I not need to clone? And just to get us all on the same page in terms of definitions, there's two different ways people make cell lines. The first is a polyclonal cell line. This is defined as a mixed population of cells that has a diverse phenotype within the population. So they all have potentially the same genetic mutation or overexpression, but it may be at varying levels. In contrast, in a monoclonal cell line, you're looking at a single cell to proliferate and create an identical population of cells that will be maintained throughout all of your experiments. There are pros and cons to each of these methods. The pros of polyclonal cell lines is that they can typically be rapidly generated, and they can be beneficial for investigating heterogeneity of response across cells. So you get a broad response as opposed to a response from something that may have been artificially selected for in your population. However, the cons to polyclonal cell lines is that lack of clonality in other cases can actually compromise reproducibility. If your cells aren't genetically stable, or if the phenotypes are not universally penetrant, it can make your data hard to interpret when you're actually using the cells in your downstream assays. For monoclonal cell lines, this downside is eliminated. This does ensure consistency of results across the assays or your therapeutic molecule production. For example, if you're making antibodies from a cell line, or you're using cells for drug screening across a wide variety of plates. But the downside of creating a monoclonal cell line is that it is incredibly labor intensive and it requires multiple rounds of single cell cloning and typically proof of clonality via imaging or other downstream sequencing methods to ensure that you have a homogeneous population. So why is cell line development so challenging? It seems pretty straightforward that after all of these years, we should have a better way to do this in terms of making the workflow straightforward. And as you can see on this picture here, it does look linear, but if you've ever made one of these cell lines in a lab, you are as familiar as I am that it is by no means a linear process and typically requires lots of optimization and many rounds of going through this process. And that's for a couple of reasons. First and foremost, the very act of editing a cell, whether it's overexpression or knocking out something in your cell, is typically very inefficient, regardless of the methodology, anywhere from 0.5% up to 20 or 30% if you're lucky. Those methods each have their own potentially cytotoxic impacts on the cells, as well as causing heterogeneity in the population. So as we talked about before, the phenotype may not be universally penetrant. Once you do get your edit of choice into your cells, you then have to create a population using one of the methods that I highlighted before. Any of these methodologies that requires you to put a cell through plastic or fluid or through a micropipetter inherently carries its own risk. Just by putting cells under fluidic pressure, you can cause cell stress to already damaged cells that are trying to survive the edits that you have done to them. And importantly, for what we're going to talk about for the rest of today, all of these methods that I've just described to you require putting a single cell in a well, whether it's limiting dilution or droplet dispensing or flow sorting. In order to monoclonize a cell line, it basically defines the fact that you need to have a single cell in the well. Cells are not meant to be alone, so typically you have an already stressed out cell that has no cellular crosstalk, and most of the time what's going to happen in those multi-well plates is you'll have a stressed dead cell that can't actually create your end population. So what I'm going to talk to you about today is our Clone Challenge grant program. And this is something that we started to tackle challenging real-world scientific problems that had failed traditional methods like the ones that I described. All of these grants came into our organization from researchers across academia, government, and private industry who were struggling to make cell lines that were necessary for their downstream research, whether it was drug screening or production. They had all tried limiting dilution, flow sorting, or other methods and had not been able to solve their problem. And so our goal in the Clone Challenge was really to use our novel technology with these grant program awardees and try to solve their cell line development needs using our tools. And so I'm going to talk about the technology a bit more at the end of the talk, but I want to just frame it for you about why our technology is so unique and what makes us different compared to any other technology. And that really the core of it is our consumable, which is a microwell array. Unlike traditional limiting dilution where you're putting a single cell in a well, we culture all of the cells that we are trying to monoclonize or create a cell line from in a single small footprint. As you can see in this cartoon here, the cells are seated in this inner reservoir. And within that reservoir, we fabricate thousands of individual microwell arrays that allow us to have spatial segregation of those cells. We can then culture and monitor those cells as they grow. And once the cells are ready for recovery, we can retrieve them, transfer them to a new 96-well plate for expansion, and ultimately for downstream characterization. So this will give you some framework for all of the case studies that I'm going to show you in the next few minutes. I just want to take a second here to highlight all of those Clone Challenge grant awardees. We are so fortunate to have had amazing applicants for this program, as well as great partners who frankly gave us access to challenging problems that we were really excited to help them solve. Just to highlight them all here, all of these case studies are available to read more about on our website, but for today, I'm just going to highlight 3 stories that I think are particularly interesting and really highlight the benefit of a new way of doing cell line development. The first of which will come from a company called Tetracore, a case study from the University of Utah, and then one from North Carolina State University. So the first clone challenge that I'd like to tell you about is the goal was really for us to help this company called Tetracore rescue their commercial hybridoma cell line. Tetracore is based out of Maryland, and they develop kits, assays, reagents, and instruments for the detection of infectious diseases, biological warfare threat agents, and biological markers. Primarily for first responders in the field, their kits can be used for early detection and keeping those first responders safe. They reached out to us because one of their best-selling kits, botulinum toxin A, had been showing increasingly poor antibody yield from their hybridoma clone. You can see here in this graph below in the bottom of the slide, in blue represents the antibody titer from one of their QC passing lots of antibody, and in gray you see the failed lots that had failed to qualify for use in their production kit. So Tetracore reached out to us to see if we would be able to help them recover the antibody producing cells from their drifted hybridoma line. And so unbeknownst to Tetracore, when they reached out to us, we had actually been trying to develop a miniaturized antibody secretion assay using our technology. And so this was a perfect fit for our technology to help solve their problem. And so really the question that we tackled with Tetracore was whether or not we could use an on-array screening platform to identify rare antibody-producing cells upfront rather than downstream in an ELISA or other protein-based secretion assay. So what you're seeing here on the right-hand side of the screen is essentially a cartoon of our miniaturized assay. We can seed IgG secreting cells. For example, in this case, we screened 20,000 bot A hybridoma cells, and we plate those cells in our consumable in the presence of a fluorescent detection reagent. So in this case, we're using a FTSE conjugated anti-IgG. And if we have positive signal, we should see a bright fluorescent signal in those particular cells. You can see what the workflow looks like here. We simply seed those cells on our consumable just by using a micropipetter. We can image the array using our technology, and then we can monitor both the expression of the antibody production as well as the growth and proliferation of the hybridoma cells. And the amazing thing about this particular collaboration with Tetracore is that we could confirm any resultant clones using their actual ELISA kits to show whether or not we had recovered antibody production. So what we did in the workflow was to perform a full array scan to identify positions that had a single hybridoma cell at day 0. You can see what that looks like in bright field here. This is just one field of view inside of the array. And these blue boxes highlight positions within the consumable that have a single cell. But what I want you to pay attention to is this one down here in the bottom left-hand corner, demarcated by a right arrow. So as you can see, we have lots of single cells. And as we follow those cells over the course of the next 24 to 48 hours, you can see that those single cells are largely proliferative, or in some cases will fail to divide, as you might expect. And so we can easily identify cells that are growing and that are derived from a single cell. But the amazing part about the assay is by doing the fluorescent scan, we can very easily and quickly identify which of those colonies are positive for antibody production using that FTSE marker. And as you can see here with the red arrow in that particular clone, we have beautiful antibody expression just from that one clone in a sea of negative cells. If we zoom in on that particular area, you can see the progression of that clone from a single cell at day 0 to an antibody secreting colony at day 1. And of course, we can then recover those positive cells and expand them for downstream testing. We can make our life much easier in this workflow by using our software to quickly identify not only single cells, but also single cells that are positive for our phenotype of choice. As you can see here, this is an example of how our software would identify a cell raft containing a single cell at day 0. We can then use the software to identify FTSE positive rafts at whatever time point we're interested in, as you can see here on the right-hand side. And we can use our software to actually overlay those two populations to identify only the single cell clones that are expressing antibody production. And overall, our software was 98% precise at identifying clonal antibody secreting cells. We were able to isolate those antibody producing cells out of the parental population. And what you're looking at here are the clones growing in a 96-well plate only 7 days after isolation and recovery. And what I think is really interesting about this particular slide is that it really does highlight the importance of understanding not only what your cells are doing, but how your cells are growing. Despite the fact that all three of these clones from A4 to G3 were in fact positive for antibody production, you can tell quite quickly that they have rapidly different growth rates, with A4 growing very quickly and G3 perhaps not having as much of a growth advantage as the others. This makes total sense when you think about the genetic drift that occurs in a hybridoma cell like this. And of course, it also emphasizes the importance of picking the right cell line for your production workflow that is both producing and proliferating. Importantly, we were able to interrogate all of the clones that we generated for Tetracore using their standard ELISA assay for this particular antibody. And we were quite pleased to find that all of the clones that we tested showed a greater than 10-fold increase in antibody specificity relative to the parental population, as you can see here in the blue bars. And most importantly, when we returned those clones back to Tetracore, they were able to use the clones to purify antibody. And as you can see in this graph, by just recloning those cells and recovering the highest producers, we were able to recover their antibody production pipeline for their commercial kit. As you can see in the graph above, that green line now represents the purified antibody relative to their early QC lot, as well as the failed lot you see in gray below. And I think the quote from Jennifer at Tetracore says it all. As she said, the purified antibodies showed remarkable improvement. They were able to get rid of all of their old non-producing clones, and we were quite tickled that they gave those new clones an ID that included the extension CM for cell microsystems. So it was a really wonderful testament to the collaborative power of the workflow that we were able to do for Tetracore and help them get their kit back into production. I'd like to highlight the success of this workflow by kind of showing you how important it is to do this at scale. We started the workflow with 20,000 cells in a single consumable and only 3 mils of media. Of those 20,000 cells, there were 68 positive events, meaning only 68 of those 20,000 cells were secreting antibody. That's 0.34% of the population. Of those 68 events, only 27 were clonal, which is 0.1% of the population. And of those 27, only 13 were able to continue growing. So from top to bottom, only 0.065% of their cell population was in fact antibody secreting and could be recovered and expanded to rescue their line. Now those numbers sound really bleak, but on the positive side of things, over 40% of the events that we observed were in fact clonal. Almost half of the clones that we were able to isolate were recoverable and could be expanded. And this is the number that we're most proud of. 100% of those expanded clones were in fact positive for antibody production greater than 10-fold. And to highlight the benefits of the workflow, from thawing the cells after they were received in our lab here to characterize cell banking and returning them to Tetracore, it was only 28 days with a hands-on scientist time of 21 hours. So if we put these numbers in perspective for this clone challenge, in a single array, we were able to recover 13 clones in 196 well plate. Using this same math, if we translated this to limiting dilution or droplet dispensing or putting a single cell in a well, in a stack like this of 16 plates, we would have recovered 1 clone. And hopefully, as you can imagine, that 1 clone that we could recover may have been like clone G4 and barely proliferated or wasn't able to survive. So again, this really highlights the utility of a scale-based approach like this, allowing us to fully understand the cells that we're isolating and using for our downstream experiments. The next clone challenge I'm going to talk to you about comes to us from the University of Utah. The lab there is working on membranous nephropathy, which is a kidney disease causing inflammation and thickening of the glomeruli, leading to kidney dysfunction. They had identified a novel antigen in MN, which was characterized by the presence of immune complexes that were limited to the glomeruli of the kidney and podocyte cells. So they wanted to make these novel mutations in a rather challenging primary human podocyte cell line. So their goal was to investigate the function of this protein by generating knockout podocyte cell lines. So they provided us with both the constructs for a CRISPR knockout as well as the human podocytes that they were hoping to generate the mutation in. And what we quickly discovered was that this population of cells was incredibly challenging to transduce. They don't grow particularly quickly, and they require very specialized conditions for growth. And when we transfected them, we found that only 1% of the population was actually expressing the GFP that was indicative of a CRISPR knockout occurring. Interestingly, when we started using our software to identify GFP positive cells, the story got even more confusing and more interesting, I suppose. What you're looking at here are two different examples of GFP positive clones. Now, if you've ever done cell sorting or fluorescent enrichment, you would know that both of these clones would come out of your sorter quite quickly, and you would have a population of heterogeneous GFP positive cells. But because we're able to grow the cells on our platform for multiple days and follow their phenotype, we can actually monitor what happens to that GFP expression. Now, in this particular workflow, the GFP, if it integrated properly and caused a knockdown, should have been constitutive. But as you can see in clone A, despite having GFP at day 0 through day 2, we lose that GFP expression, suggesting that it was not stably integrated and the cells were not knocked down. In contrast, in clone B, you can see that fluorescence intensity is maintained throughout the culture period, and we can successfully isolate these cells. So to me, this really highlights the import of an imaging-based system like this, where we can use our software and our instrument to monitor clones that are stably integrated, even if they are low frequency or rare events. Now, overall, we were able to perform clonal expansion of these stable GFP cells. Less than 0.05% of the screen cells had stable GFP expression. You can see one of the example clones over here on the right-hand side, that as it expands out from its growth surface here in the middle, the cells are in fact GFP positive. Over 50% of the isolated monoclonal stable GFP colonies expanded, which translated to 0.003% of all the cells screened. So again, finding that needle in a haystack with any other method would be nearly impossible. We were able to deliver 4 clones from transfection to expansion to our Clone Challenge awardee in only 18 days. So the last Clone Challenge I'm going to talk to you about today comes to us from North Carolina State University, and I like this one a lot because it really highlights the challenges of cell line development, as well as the role of both 2-dimensional and 3-dimensional culture in these types of assays. So this lab at North Carolina State University is studying Angelman syndrome. It's a neurodevelopmental disorder characterized by intellectual disability, ataxia, and sleep disorders. And this is primarily due to the loss or loss of function of the UBE3A gene in neurons. So the goal of the lab at NC State was to generate a reporter gene knock-in that was into patient-derived iPSCs from Angelman syndrome patients for use in drug discovery and screening for UBE3A reactivation. Now, the challenge here was quite unique. Not only was their reporter gene knock-in enormous, but the cells were very difficult to transfect, and you could only see expression of the reporter successfully after the cells had differentiated. If you've ever differentiated neurons in either 2 or 3 dimensions, you know this is typically a 3 to 4 month process. So it's a pretty labor-intensive workflow to get all the way to the end and potentially not have the mutation or the expression of your reporter. So they came to us to work in our Clone Challenge program to help them derive their monoclonal patient-edited cells. This was a slightly different workflow for us due to the difficulty in transfecting these cells. We took a pooled approach to start with rather than going right to monoclonization in order to maximize our chances of success. We electroporated their proliferating iPSCs with their CRISPR-Cas9 constructs. And rather than monoclonizing, we created polyclonal pools for expansion. Using genomic DNA PCR of those polyclonal pools, we were able to confirm that of all of the pools that we tested, there were only 2 positive pools in the population. However, we could then take those pools and rescreen them on a different consumable to create monoclonal cell lines, giving us a much higher possibility of success. So we took the pools, monoclonized them, and then isolated cell rafts containing monoclonal iPSCs into our downstream 96 well plates. As you can see an example here on the left-hand side, here's a single iPSC that starts to divide and form a colony, and we see beautiful iPSC growth in only 8 days. After the clones were expanded, we were able to confirm their edits using genomic DNA PCR downstream, as you can see on the right-hand side. And of everything that we tested, we were able to achieve 17 edited monoclonal cell lines. Again, that sounds like a huge number, And it was, but the important thing here is because they had a very specific downstream phenotype they were trying to achieve, they wanted to be able to test multiple edited lines to determine which was the right one. Importantly, after we generated these iPSCs, both we and the lab at NC State differentiated the cells. You can see on the left-hand side, the iPSCs were differentiated into mature neurons in 2 dimensions. And as you can see in the bottom 2 panels, in GFP, the region of interest that we knocked in as a reporter is in fact expressed in the reporter, but not in the parental. In addition, we were able to use the cell raft array and the cell raft air system to create 3D organoids using the iPSC line. You can see what that looks like here. This is a 10-day-old early differentiation into a cerebral organoid, and that cerebral organoid is in fact positive for our GFP reporter. After further differentiation off array, the lab at NC State took those organoids and cryosectioned them and stained them for neuronal markers, as you can see here with TUJ in red, and our GFP reporter in green, which you can see here in panel B. So in this workflow, we were able to generate patient-derived iPSCs with a specific knock-in that could be visualized upon both 2-dimensional and 3-dimensional differentiation. So what we learned from this clone challenge was that we didn't always have to get it right the first time, that this 2-step methodology was very useful for incredibly challenging cells with a low penetrance. This workflow allowed us to generate 17 monoclonal candidates and then create this patient-derived reporter line that was capable of both 2D and 3D differentiation. This was a really fruitful collaboration for us, and the lab actually recently published this work into a peer-reviewed manuscript, and they presented it at a national meeting. And this quote from Sam, the grad student on the right, is my favorite of all time. She said, "I'm still in disbelief, we did it, still crying tears of joy." And there's nothing better as a researcher than being able to help a grad student move their science along in a way that they weren't able to do before. So of all the clone challenges that we have tackled through this program, we have had a 100% success rate delivering the desired cells, irrespective of whether or not the phenotype is rare, or there's been a rare edit, or it's a challenging cell line or problem. Importantly, after giving those cells back to the labs that contacted us, they have been able to verify clonality and phenotype in their own labs, confirming that we were able to deliver them what they needed. And importantly, for these researchers, we were able to complete the projects in weeks, not months or years. So what have we learned overall about improving cell line development? So first and foremost, single cell survival overall can be improved dramatically by keeping cells in culture rather than isolating single cells. Another key takeaway here is that scale matters. For rare phenotypes or challenging edits, having hundreds of clones will ultimately provide you a more opportunity for success than relying on survival of the fittest, especially in cases of genetic drift or instability or rare phenotypes. If you're limited to the one clone that survived, it may not be able to recapitulate the population that you're looking for. We've also found through the podocyte example that image-based clonal selection can improve your phenotype stability because we're able to upfront choose the cells that we think have the best chance of being edited rather than relying on cells surviving without that forward screening. And importantly, we like to say that no cell line is unclonable if you have the right tools at your disposal. So what are those tools? I'm sure by now you're curious about how we actually do it and how we could help you do it or how you could do it in your own lab. We do this using our technology, the cell raft array, which I've already highlighted several times for you, and our instrumentation, the cell raft air system. You can see what this looks like here in this cartoon. It starts by simply getting the consumable ready for cells. And all you need to do is prepare a cell suspension that can be deposited into the inner reservoir of the array. The whole process is very quick, takes about 15 minutes of hands-on time. Once the cells have attached to the consumable and are in the array, we can perform image acquisition in bright field and 4-color fluorescence using our instrument, the cell raft air system. Imaging is fully automated and very rapid. Typically, scans take between 5 and 10 minutes, and they can be performed over multiple days or weeks depending on your experiment. You can see what that looks like here. I've shown you some examples of this through the talk. But importantly, we're able to generate a complete track and trace growth record of your cells from day 0 all the way to the end of the growth period, regardless of your starting point. This gives us superior survival of our single cells because even though the cells are single cell segregated, they're never actually alone and they're sharing contiguous media, making it a much more hospitable growth environment. We can perform rapid imaging in 4 colors. And again, we can create that track and trace growth record to give us confidence of monoclonality and phenotype. Once we have generated all of that data, as I've shown you previously, we can use our software to perform image analysis for a variety of criteria that allows us to perform clonal selection for downstream expansion. What this looks like in our software is that we can very easily use the software to identify single cells at the time point of interest, as you can see here in the green boxes. We can use our software to measure growth rates if we're looking for a specific doubling time or a specific or a specific confluence before isolation, as you can see over time in these particular rafts. And then we can very easily characterize phenotypes, whether it's GFP expression, antibody secretion, what have you. We can do that using our software to help us select. And then as you can see in that overlay between the Venn diagram, this is our sweet spot. These are the clones that we actually care about. And it allows us to very quickly and easily select high quality clones for isolation. Now I've said isolation quite a few times in the last half hour or so, and I'm sure you're curious as to what that actually means. So the key here of the technology, really what makes the whole operation tick, is that each of those microwells contains a fully releasable cell growth surface called a cell raft. It's comprised of polystyrene, and so it's no different than any other cell culture surface that your cells have already been accustomed to. The raft is completely releasable from the microwell array using our instrument, the cell raft air, which essentially dislodges the raft and transfers it over to a 96 well plate. You can see what the instrument looks like here. Very straightforward operation. With the air system, what you see is what you get. All you have to do to turn it on is push that button on the left-hand side. It's compatible with any 96 well plate you already have in your lab. It is completely fluidic and enzyme-free. There's no sheath fluid, no plastic tubing, absolutely nothing to disturb the growth of your cells. And that makes it very easy to operate and use and maintain. Very different from a sorter or a dispenser where you need some specialized knowledge to get started with it. You can just turn on that button and get started with the air system almost immediately. The other nice thing about the air is that it's very flexible. You could isolate 1 raft or hundreds. The choice is yours. So you really can scale the workflow to your needs and to your research. Once you have isolated the rafts containing your cells, they simply get maintained off of the array for the duration of the growth period that is important to you. They go right back into normal culture without you having to change your workflow at all. And again, it's important to highlight that what we're isolating is the raft itself and not the cells. So we're actually isolating the cells that are attached to the raft, which means your cells are already past that initial single cell growth phase and will get back into log phase growth much faster. You can see what this looks like here as this video plays. This is an example of a colony that's been transferred. The raft is sitting in the left-hand corner of the screen. The cells will simply begin to grow off the raft and attach to the floor of the new 96 well plate. This happens totally passively, completely hands-free, and eventually that raft will get dislodged and be dissociated and will be aspirated during normal culture. So what does this mean for you in the lab and for our Clone Challenge winners? So what we were able to deliver for them was a full plate of validated clones. You're not putting a single cell in a well and hoping that it survives. You're actually cherry picking your targets and ensuring that you have the best chance of success. It also means you don't have to feed empty wells because everything that you've picked, you've already assured that it's growing up front. We get rapid outgrowth because of this, and ultimately you have plates full of what you need with no downstream consolidation. I've shown you 3 examples of particularly interesting or challenging cells in this webinar today, but we have performed this same workflow with hundreds of cell lines at this point, both in our lab and with our customers. We have that information publicly available in our cell line atlas, which helps you get started with our technology, either in my lab or in yours. And if you don't see your favorite line, let me know. We're always interested to figure out what we can clone next. We are fortunate to have a really wonderful group of collaborators and customers. You can see here just a brief list of some of our customers who've adopted this technology in their own labs and have been using it to do celling development and other workflows. If instrumentation is not in the cards for you right now, or you're really just curious about how you can use our technology to get your own lab started quickly, we also offer lab services at Cell Microsystems. We offer custom cell line development as well as bespoke assay development. We can help you accelerate your data generation and maximize your team productivity. And ultimately, when you decide that the air system is a good fit for your lab, it gives you a seamless transition to ownership because we will help you get started with all of the protocols and assays that we've developed for you. So if you're interested in lab services, that is an option that we provide as well. If you are interested in learning more, our website is listed here, as well as the ability to contact us to talk about demonstration of the technology. And as I mentioned at the beginning, all 10 of the clone challenges have posters or application notes associated with them. So if you're curious about going more in depth in any of the data, it's accessible on our website, where you can let us know in the comments, and we're happy to send it to you directly. So with that, I'm going to close, and I'd like to thank you for your time, and I am happy to take any questions that you might have.
Thank you, Dr. Hartman, for that fantastic and insightful presentation. I loved all the data, workflows, and real-world applications. It was very cool. Before we jump into Q&A, we'd love if you can answer a quick poll question. And thank you in advance for taking a few seconds to respond. I just launched it, should be showing up on your screen. And while people are answering that question, we will dive into Q&A. Don't forget to submit your questions into the chat box. And we have a few questions already asked to get started. For monoclonal cell line development, how many clones do you recommend?
Yeah, that's a really good question. That's very much dependent on your downstream. We have some labs who only want 3 or 4 clones, and we have some labs who want hundreds. It's really dependent on the scale, how rare your phenotype is, and what your capabilities of downstream analysis are. So some groups have lots of robots and high throughput PCR machines, and they want to characterize lots and lots of lines, in which case you could isolate hundreds, potentially thousands. Or if your phenotype is fairly penetrant and not rare, you can isolate only as many as you need. I will say, typically speaking, most of our customers and our clients, we isolate between 1 and 2 96-well plates. Typically gives you way more of the clones than you need in order to get the phenotype that you're looking for.
Okay. What concentration of cell suspension is required for the initial plate array?
The nice thing about the array is that we need very few cells to get started. Typically in some of these workflows, when you go over to the cell sorter, you wouldn't even dare book sorting time without 500,000 or 1,000,000 cells. For the most part, in our largest format consumable with the most number of positions, we seed anywhere between 30,000 and 50,000 cells. And we could seed as few as a couple of hundred, depending on which array type you're using. So that's really a nice feature. You can start screening far earlier than you would in other potential technologies, and you don't need as many cells to expand, potentially leading to genetic drift or instability.
Okay. How long does it typically take to go from seeding the rafts to being able to transfer them to a 96-well plate?
Typically, I would say most experiments, it's 3 to 5 days, depending on the growth rate of the cells. For the most part, you typically let the cells get to the 4 to 8 cell doubling stage, depending on your needs. You could isolate theoretically at the single cell stage. If you really just wanted proof of clonality and you wanted a single cell, you could certainly isolate at the single cell stage. Or if your cells were slow growing, it might take you, upwards of a week to get the cells to divide to a sufficient level. But typically we find that if the cells get to that 4 to 8 cell stage, we have a very high success rate of them continuing to grow after isolation.
Okay, and the next question is, what is the largest raft size that you offer?
Sure. The largest raft size it corresponds to that 3-dimensional workflow that I briefly highlighted in the NC State Clone Challenge, which is a 500 micron raft. So they come in 3 sizes, 100 by 100 micron square, 200 by 200 micron square, and 500 by 500 micron square. So we use those 500s for a 3D culture of large organoids up to about 1 millimeter in diameter. And you can select the 100 versus 200 rafts for your cell culture workflow, depending on how large your cells are, how many cells you want to grow, etc. Of course, the number of positions that are available to you is proportionate to the size of the raft. The larger the raft, the fewer the positions.
So how many, which cell raft has the most wells in it?
Sure. The raft with the array with the largest number of rafts available is our 100 single. So it's a single reservoir with 100 by 100 micron rafts, and there are 40,000 individual growth positions in that footprint, which requires only 3 mils of media to grow.
Okay. So that's all the questions we have at this time. As we wrap up today's webinar, I want to extend a huge thank you to Dr. Hartman for sharing her expertise, and to all of you for spending part of your day with us. Your curiosity and questions make these sessions very valuable. Before you go, please take a moment to complete the brief survey that will appear after we end the discussion. Your feedback helps us make each webinar better than the last. And remember, a recording of this session and additional resources will be emailed to you soon and available on our website. Thank you, everyone, for attending.
Thank you.